manager. You guys know about the package manager, the Conda package manager. Okay, you don't have the link. So yeah, we have shared the link. You can access from link also. So first of all, we will need to install the Conda Collab. So you guys can execute the first code chunk by pressing to this play icon. So you can see it's executing here. Is it visible to everyone?
Yes, sir. Yes, sir.
Okay. So now we have installed it. So we can run this next command to connect our terminal to the Conda. So just click on this play button and it will connect it to the terminal, to the bash module. Are we done?
Yes, sir. Yes, sir.
Now what we'll do, we need some space. So Collab also provides us a storage, but it is for temporary use. We need something permanent to store our files, which will be generated during our analysis. So what we'll do, we just go to this toolbar and we will click on the files icon, and you will see an icon here showing mount drive. I have already mounted, so it's showing unmount drive. You just need to click on it, so it will mount your Google Drive onto the Collab.
Sir, excuse me.
Yeah.
It is showing your session crashed for an unknown reason after installing the conda.
Okay, your session
Crashed.
crashed. So is there any option to restart it?
Sir,
Is it
I'm doing
showing?
it, but it's showing crashed.
Okay, so you can close this. You can close the Collab and reopen it. Try that.
Okay, sir.
So once you have mounted the drive, just go to this terminal option and click on it. The terminal will open. Let it connect.
Sir,
Yes.
so mounting can be done by only clicking on this icon.
Yeah. Excuse me. So are we done with the drive mounting?
Yes, sir.
Okay. So What you have to do now is your terminal is open, since you just have to select this command from here, source tilde slash dot bash rc, and right click on it. You will see copy the selection. Don't use the copy option. It will copy everything in the code chunk. You just need to copy the selected part. So copy selection, go to the terminal, click on it. You can either paste by right clicking, or you can do Control Shift V.
Excuse me, sir. It's still showing that it is crashed.
Okay. So anybody else facing the same issue?
Do it require any kind of special permission or something?
Same thing.
Yes, it's saying your session is crashed.
Okay. So refresh it as other people are suggesting. Okay, try refresh it. Uh, is it solved? Is it working now after refreshing?
Yes, sir. Now it's working.
Okay. Thank you for the suggestions. So you will paste this source tilde slash dot bash rc and hit the enter. So
Done, sir.
yeah, so everyone with me?
Yes, sir.
Yes, sir.
Okay, so now you can see this bracket base, bracket close. Is it showing on your terminals?
Yes,
Yes,
sir.
sir.
Base
Okay,
content.
so this means your terminal got the access of Conda. So this base thing means you are in the base environment of your Conda manager. So now what we need to do is we will just go into the drive directory. So we'll do ls, enter. You will see a sample data directory, a drive directory, and conda collapse install.log. Even if you don't see these conda collab install.log or sample data, it's okay, but you should see this drive directory there. Is it visible?
No, sir.
Yes, sir.
So
Yes, sir.
if it is not
Um,
visible, this means your drive is not mounted. Check again, please. So we'll move into the directory by cd drive, enter. So we are now into drive. We'll do ls. We'll see my drive and shared drives. We'll go into the my drive and press enter. So everyone is with me, or any issues?
sir, just a minute.
Yes, sir.
Okay, what is the issue, Swapnil? Again, session crashed. So it might be the network issue, I think. Is your network stable?
No such file in directory.
You can refresh and rerun the commands. Okay, so we'll do ls again. Check what files you have there. These are my files. You won't see these directories and files there. If it is empty, no worries. It's okay. It's totally fine there that you don't have anything.
So, sir, can you repeat after mounting?
After,
Mounting the
okay.
drive.
So once you have mounted, you just need to paste the source tilde slash dot bash rc by just selecting it and uh, 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 right clicking on it and copy the selection and paste it to your terminal and then hit enter.
Yes, sir. And sir,
Okay.
after that, let
Uh,
CD
you got
drive.
you got the base thing.
Yes,
Okay. Now you just have to do ls. Check whether you have the drive directory or not.
yes, sir. Base content and drive.
Okay, now just go inside that using cd command, cd drive, then do ls. You will find my drive directory. Then you will do cd my drive. We are just moving into the
Sir,
drive.
I have done CD drive. It
Yeah,
shows no such file or directory.
there is no my drive.
I, no, I had done CD drive, not my drive. I had done CD drive.
Yeah, cd. Uh, can you provide a screenshot?
Yes, sir. Yes.
Okay.
Probably her drive isn't mounted on the collab node.
Yeah, that might be the case. You can also check whether your drive is mounted or not from this file section. Just go there and you will see if it is mounted. You will find a cross here, like not cross, a slash. That is cutting this icon.
Sir, I have
If,
sent the screenshot. Can you check?
okay. Hmm. Okay, so you are executing my drive before executing CD drive. So first you need to go to drive. So you are in drive. Do ls, please.
Yes, sir. I have done LS. It's showing base, bracket base, then space slash content slash drive hashtag. Am I audible, sir?
Yeah, you are audible. Uh, but you are in drive, then there must be my drive. It is also showing in your previous commands. Then it should be there. Can you see my drive after executing ls?
Yes, sir. Sir,
So
I had
you
done cd my drive slash and enter.
so you are now inside the my drive.
Yes, sir.
So it's fine. You are on correct track.
Okay, okay, sir. Sir, after that,
So now you are inside that. So again, I'll repeat that if you don't find any file in my drive, that's totally fine. You just need to make a directory named workshop. So what we'll do, we just type mkdir space name of the directory workshop, and you will hit enter, or you can name it anything you want. Like I'm making work only because I have already workshop. So yeah, are we done with this?
Yes, sir.
Okay. Now you just need to move into the that directory. Do cd work or whatever name you have given to that workshop. You should see the proper address, content, drive, my drive, and your directory name.
Yes, sir.
Okay. Okay. So now we are going to create a Conda environment which will handle our tools and let us install them in a specific address or specific room. Okay, now you just need to copy this command from Conda to FastP. Copy, select it, copy the selection, and paste it. Sorry. Wait. No. Yeah, we are doing right. Sorry. Just paste it. Copy, Control Shift V. Paste the command and hit enter. It should be something like this. Your interface should be something like this. Should
Yes,
show collecting package metadata.
then
Okay, it will take some time. So what we are doing here, we are creating a Conda environment which is named skill underscore env1. So Conda creating, since we are creating, so Conda create dash n is for the name. We are specifying the name of the environment, skill env underscore env1, then space, we give y is for yes. It will ask a question that whether we want all these to be installed, or whether we should replace, whether it should replace the already downloaded packages. So we will ultimately, we are going to do yes. So we have given y here, and then c dash c space bioconda, which means we are using the channel, c for channel, bioconda, to download SRA tools space fastqc space multi-qc space fastp. So fastp is the tool we are going to use for trimming the sequences. So we are on the same page or not, everybody?
Sir, it's taking some time
Yes, sir.
for processing.
What?
So it is taking some time.
Okay, okay. Let
Currently,
it
it is not preparing transaction.
okay, let it be. Until you see this address line again, let it run. So we have installed the SRA tools, which will help us to download the raw data from SRA database, as I have mentioned in the theoretical part, and then fast QC, which will generate the individual reports, and multi QC for the aggregation. So we are not going to take multiple samples. We'll take only single sample, but we can execute multi QC to check single individual sample also. I will just show you how to do it. You can execute multi QC on multiple samples also. So can we move forward?
Yes, sir.
Okay. So once everyone is up until here, what we'll do, we'll just copy this command. What it says, activate the conda environment using the below command. Okay. So we have created an environment called the skill underscore env1, and now we are just activating it. So just copy the selection and Control Shift V and hit enter. So when you will do this, you can see that the base is replaced with this environment's name.
Still involved.
Okay, so this means you have activated the environment. Now you are into that particular environment, that particular room, which has SRA tools, with
জগন্নাথ
the fast
কখন
QC,
ছিঁড়ে দিল
multi
গাল
QC, and the fast P.
Yes, sir.
Now what we are going to do, we are going to download the FASTQ files. So for that, we need to execute this command, faster qdump, and the accession ID of the SRA data. So this is so we are doing analysis on a already submitted files. That's why we are going to download it. When you have your own files, your own data, you don't need to download it. Of course, you will not need to do that. You can simply execute fast QC on that. So uh, for this session, we are downloading this particular organism sequences, raw sequences. This ID belongs to Mycobacterium tuberculosis bacteria. You can also search it on the web to know more about this. So we'll just uh, copy this command, copy selection, and Control Shift V. So faster queue dump, it will collect collect the data from the SI database, and there will be only a single binary file on the database to make it more convenient for the database organizers to store it. So what do we need to do is we have to use the faster queue dump, so it can collect that binary file, and then we use split files. Since it is a paired end sequence data, that's why we need to use split files into two different files of paired end sequencing. So double dash split dash files, and then E is for threads. uh, I think you guys know threads. And M is for the memory. We can also skip the memory and this part. You guys don't need to do that. You can just copy and paste. And this double dash temporary space slash TMP. So TMP is signifying temporary directory, which will it generate in Collab for storing the in-process data into the TMP directory, which will be deleted after the execution is completed. So once you are done with that, you just need to hit enter, and it will download the data for you. So everybody executed the command. It might take some time. Hmm. So anybody has any issues up until now? Any errors or anything you didn't understood, want to clarify?
Sir, actually my collab was not working, so at that time I couldn't do that.
Yeah.
Sir, matlab, I was doing this. Uh, it has some error, like paired end वाला जो command है उसमें लिखा आ रहा है called process error. Command not found भी आ रहा है उसमें.
Command not
S
found.
is paired end reads from SRA database वाले में.
Okay. Command not found. So your environment was successful, no? Your creation of a environment was successful or not?
Yes, sir.
Then it should. Can you share your screenshot? Okay,
1 minute,
so
sir.
okay, Shivangi. So splitting we are using since the SRA database stores it in a single binary file. So if we won't use it, it will merge everything into a single FASTQ file, and that would be problematic when we use it for downstream processing. So that's why we are using split files to like tell it to just split the files. It has its it has its algorithm to do so. And the thing is the threads. Threads are basically the cores of your computer. So what happens is when we run a command, it occupies a particular what we call it node. So in your system, suppose you have a 16 core or 32 core system. So it's basically the hardware and software thing. So it's for computation for computers. So I might not know that much, but uh, what happens when we execute a command? It occupies a single core. If we will execute the command without specifying thread, that is -e, and when you specify how many threads you want the command to use, it will occupy that many threads. Like we have specified 4, so it will occupy 4 threads and will execute the command parallelly. So it will be much faster and easier for the computer to process. Did you get that? Okay, so everybody has executed the command. Can you see these uh, spot read equals to uh, 2,300,000 and 700 something something. These are the number of reads. So this many reads.
Sir,
Yeah.
sir, mine is showing error. I don't know what happened.
You can share the screenshot, please. So now you can check that whether your files are there or not. You just need to do ls, hit enter, and you can see that you have downloaded your raw sequencing data. You can see er something something underscore 1 dot fastq and underscore 2 dot fastq. This is the accession ID. Yes, command. Uh, okay, what's in it? Yeah, you are doing right, Sweksha. Why did you uh, press Control C? That kills the command execution.
Sir, actually, it's by mistake. That's
Okay,
why I asked.
no problem, no problem. Uh,
Sir,
you
am
can,
I on right track?
yeah, you're right.
What
Okay,
should I do?
wait until you can see these stats on your screen. Spots read equals to this numbers, and read equals to this. So you can
Sir,
see.
I got it. I get it.
Okay, now so you are with us. Now you can check it by executing ls command that your files are there or not. So everybody with me. So as we have downloaded our sequence, now we can move on to the fast QC step. So what we need to do is, for fast QC, to execute fast QC, first we need a directory where the fast QC tool will save its results file. So this is required for fast QC, that we make a directory before executing the command. So we just you guys just need to copy paste it from here, or you can generate by execute typing the command also. This thing is mkdir. Best is in this.
Done.
So you guys made your directory. Now you guys just need to copy this command, copy selection, and paste it and execute. Okay, I have misspelled it, I think.
And your directory name is like sample, and then you have put
Underscore. Yeah. Okay, just need to remove the directory. Thank you. Okay, so you guys can see this interface is it's showing approx 5% complete.
Yes, sir.
Okay, good. Okay,
Sir,
so Apnil,
sir,
you are running the command from the code block. You don't need to execute that from. You just have to. Okay, don't run. Yeah, don't run on the collab. Just execute the command on the terminal. Copy and paste it. Okay.
sir, can you see my screenshot?
Yeah.
Sir, I send the screenshot. Can you please check? In mine is showing something like that.
Duplication failed due to. Can you?
Sir, I'm sending again full screenshot. Yes, sir, I sent.
So your command is executed successfully.
yes,
It's
sir.
okay. You can just check whether there are files in your fastqc directory or not. Okay, so Apnil,
I'm
after
sorry, I did not understand.
you just need to go into your fastqc directory,
Mukul
cd.
Sveksha is running fast P instead of fast QC.
Oh,
Here come.
she ran it over that. I'm sorry.
What?
Sweksha, you need to execute fast QC, not fast P now. We are checking the quality before executing trimming. You have trimmed the reads. So we ultimately we are going to do that. So you can now execute fast QC. Just copy the command from the code block. Okay.
Sir, I have given the wrong command. Really?
Yeah. Not wrong, but you are ahead of us. just go back and execute the fast QC command. Okay, this one, fast QC, the file's name and then output directory. So everyone, you have your execution completed. Can check uh, by doing cd fast fast qc sample1, enter ls. So you can check that you will get 2 HTML files and 2 zip files. So zip files are FastQC binary files, and HTML files are your reports for each of the sequence files.
Sir,
Yes.
after executing that fast queue and that code wala file, we have to do the ls.
After what? Yeah, FastQC.
After
After your FastQC is completed, you have to check whether your files are have generated or not. They will be saved into your FastQC sample1 directory. So just go into that directory and check that these 4 files are there or not. So are we done with FastQC? So did you guys get these 4 files? Now you can come out of the directory by doing cd space double dot enter. You're back into your working directory. Yeah. Now we'll execute the multi QC command. Just need to select the multi QC command, copy it, and Control Shift V. Press the enter. So we have generated our fast QC reports. Now we are just aggregating these 2 reports into a single, more comprehensive, more interactive uh, multi QC report. So you can see something like this, and you can check that this has generated a multi QC slash so underscore sample1 directory, as we have provided in the command multi QC. For executing multi QC, then fast QC underscore sample1. This is where it will take the files from. So suppose you have multiple samples. So what we will do, you will save all the fast QC report into a single directory. Here we have fast QC underscore sample1. In this directory, you should save all your fast QC reports, and then you will execute your multi QC command on that particular directory, and will assign -o the output directory's name. You just need to assign the name here in the command, and it will generate its own uh, directory.
sir,
Yes.
can you please take the screenshot in my
Okay, see. So did you executed the fast QC?
yes, sir. I will get to that.
Not executing it in the directory that she's supposed to execute in, like in the work directory. She's one directory back in the my drive directory.
Okay.
And doing the trimming first does not matter because here the files we are giving different files, I guess.
Okay, you just need to go into your directory again. Go into your work directory. Thank you for suggestions.
Sir, sir, what?
You just need to go into your work directory again. You have came out of it.
Okay, okay.
Yeah, work and workshop. Workshop. Just do CD workshop and you will be inside that. Then execute the multi-QC command. Okay. So now we have the fast QC and multi QC reports. So these reports are saved in your work, this Google Drive. You can just go into your drives and download it from there, and then we can uh, analyze them in a browser. So just go to your drives, respective drives. So I'm in my drive. Here I will find the work. I will go into my work directory and I can
Sir,
see. Yeah.
sir, should I execute from starting?
No. You just need, you have your fast QC files ready.
It's not showing actually.
Have you executed fast QC or not? Can you go into your fast QC directory, fast QC underscore sample one directory, and do ls and tell me how many files do you see there? Other people follow me here. We are in our respective drives, and we can see these two directories here also. So you can go into this and you can see these two for each of your files. Here is your fastqc HTML report. You just need to download it first. Download into your computers.
Sir, it's showing it's not exist.
Okay, not exist. So you your fastqc is not fastqc command was not successful. So you can execute the fastqc command again. So once we have downloaded the file, you can open it into your browser and you should see something like this. Is it visible to everyone?
Yes, sir.
Okay, so you can see your fastqc for trim one. Sorry, this is not the file. I'm sorry. Mm, but
Sir, I share the screenshot. I unable to get.
okay, we'll check it. So everybody can see this Uh, so your fast QC report is on your screen. So what parameters we can see here? First metrics is simple. These are basic statistics. The file name, what we have downloaded. Then the file type, it's a conventional base calls encoding. So it is either from Sanger or Illumina. It has detected this. So it is from Illumina. These are Illumina sequencing reads. And then total number of sequences, how many sequences are there? There are these many sequences. Then total number of base pairs. Then so sequences flagged as poor quality. So you can see that these sequences has 0 poor quality uh, sequences tagged. This means this data is sequenced very accurately and very nicely. So there is no need to trim, but we will trim it for the demonstration. And the sequence length is between from 35 to 151. The GC percentage we can see here is 65. So you can see this in front of each matrix, there is this right sign and cross and exclamation. You can see these 3 types of uh, indications. So right means this matrix is good, and cross means you need to do something about this, and exclamation means it is okay but can be improved. Okay, so first the basic stats, then per base sequence quality. You can see these box plots. So most of your reads are falling above your uh, 28 quality score. These are on y-axis. You can see quality scores, Q scores, the Fred score, and on your x-axis you can see the position in reads. Okay, so the read is average read length is 151. So most of your reads are lying above 28. So we can consider that our sequencing is been done nicely, and these are showing very good quality. Now second is per sequence quality score. So for each sequence we can see here these on y-axis we have number of sequences, and on the x-axis we have mean sequence quality, average sequence quality. So you can see that most of the sequences are of greater than 30 Fred score. So again, the sequences are of good quality. Now uh, we have per base sequence content. So there is some problem, as you can see this indication. So most of the times when you have a good sequence files, you should see almost parallel lines for each base pair. So these different lines for of different colors, you can see each represents the line for the content of a particular base. Okay, so for T it is red, for C blue, for A green, and G yellow. So you can see some disturbances in starting and somewhat at the end. So it is also fine. It's not that much of a issue as uh, in when we call a sequence bad, if these peaks fall above 80 or 70, then we have some major issues. Then we need to cut off that regions out of your reads. So it is okay. It's not that bad. and then per sequence GC content. So as you can see, this peak is formed. So the blue peak is theoretical distribution, the normal distribution. It should be like this, and our peak is red. So it is above the normal distribution. So it is showing some little bit issue. It's okay as we cannot see multiple peaks here. Uh, if we would have analyzed some RNA-seq data or something else, we assume that we would have got some multiple peaks. And if you get multiple peaks in a isolated data like this one, then that is an indication of contamination. So we have one peak, which means our sequence is clear and there is no contamination, and it is very much good. Then we have per base n content. So how many n's are there in our sequences? So you can see there is almost no n bases in our read sequences. Further, we have duplication levels. So we can see that most of these sequences are single, as we have duplication level on the x-axis and number of, or the percentage of total sequences on the y-axis. So it is falling. So it is also again a good thing. Then we have sequence duplication levels are done, overrepresented sequences. So you can see there are no overrepresented sequences, which means the adapters are not there. No remaining adapters it could detect. And then finally, the adapter content is also 0, and overrepresented sequences also show the sequences which are repeating itself in very high numbers. So there is no such sequences. So it is a very nice and very high quality data. So we can process it and we can directly use it for assembly. But for demonstration, we'll do the trimming part, and you can also analyze the other FastQC report. So this is for individual purpose. If you have multiple samples, then we have to analyze the multi-QC report. We can also see the multi-QC report. Let us check that also. You can go to your respective multi-QC directories and then download the multi-QC report. Once it is done, you can see it from here. Open it in your browsers and just you can see the interface. Okay, so it is much better than FastQC interface. It is more interactive, as you can see, and you can see the multiple samples in a collaborated manner. So this is your first file and this is for your second file. So you can see the duplication percentages 27.6 for first one, 25.1 for second one. The GC content is similar, so we are good to go, and 2.3 million in each file. So we should have equal numbers of reads in each file, each paired in files. Okay, so duplication is not an issue here. And then you can see that sequence quality histograms, these are also good, above the 30. Some issues are at the end, we can trim it off. Then we have per sequence quality scores that are also good, not any issue about that. Then we have the per base sequence content. So it is a heat map. You can click on it and it will change into your particular files plot. So this is similar to fast QC. Now back to heat map. This was for first one, and this is for second. So you can check almost parallel uh, some disturbances at the starting and at the end. Then we have the GC. You can see both of the files have almost the same peaks. So this is also good. Per base n content is also good. Some disturbances are here, but that's fine. This is not much. Above there is a threshold. So if it is greater than 1%, then that there is some issue. Then sequence length distribution. So we will remove the shorter sequences out of this using the trimming tool. So it will become okay. Then sequence replication is also not bad. It's good. And then adapter content. So adapter content is almost 0. So yeah, if it is more than 1%, then there is the alarming thing. So at the end, we get a final heat map of the summary of the sequences. So you can see there is some issue with the per base sequence content, but when we trim it off, it should get away. Okay, so this is your multi QC. You can also go and read about these modules. Yeah, it has the references also present here, and we have also a tutorial video. You can go and watch it for better understanding. Okay, so this was the multi QC. Next we are going to do is trimming. So anybody has any doubt up until now?
Sir, can you please explain the GC content?
GC
The,
content. Okay.
the peak, the peak.
Okay, so for us, for an individual sequence file, you should get a single peak. Okay, and it should be normally distributed. It should not be disruptive or directional.